Hi everyone and welcome back to Cancer Q&A. This episode will conclude our video series and we are so grateful that you have all joined us to watch this series.
In our last episode, we discussed 3 of the 5 genes that we researched, RAB3B, KNTC1, and RIMS2. Today, we will be talking about the remaining 2 genes, MCAM and DSG3. Both of these genes are cell adhesion molecules, meaning they aid in keeping cells together and help with communication between cells that are next to each other.
To start the research on each gene, we looked through papers in scientific literature to see what other results people have concluded in regards to how these genes may impact cancer survival rates.
MCAM, which stands for melanoma cell adhesion molecule, is the first gene we're going to be talking about. We wanted to cite a paper about how MCAM is a great marker of metastasis and how it can play into apoptosis and spreading of cancer. In this paper that we read, the scientists remarked that MCAM was indeed present in abnormal amounts in tumors. And this makes sense because cancer cells like to cluster and grow in strength, meaning having a cell adhesion molecule to keep the cells together would be helpful for the cancer to enlarge and spread.
To test whether MCAM significantly altered the ovarian cancer's ability to grow, the scientists silenced MCAM in cancer cell lines. They discovered that this silencing enhanced apoptosis, or programmed cell death, of the cancer cells. This means that MCAM was a contributing factor in keeping cancer cells alive and strong. In addition, they discovered that tumor invasion was also reduced. This makes sense because if there is less expression of the gene controlling the sticking of cells together, the cells cannot cluster and grow as much.
After reading this paper, we wanted to see if we could reach the same conclusions with a bioinformatic analysis with the gene. You can see here in our graph on CBio portal that those patients in blue with low expression of MCAM had a higher % survival in the early months as compared to those people in red with high expression of MCAM.
We also looked on KM plotter to check our results. In this graph, we focused on patients with triple-negative breast cancer and an extremely aggressive type, and found again that those with a lower expression of MCAM had a higher survival rate as compared to those with a high expression. This graph was also statistically significant with a p-value less than 0.05.
This means that MCAM is most likely a gene that aids cancer and growth and development.
The next gene we are going to be talking about is called DSG3, and the inhibition of DSG3 has been seen to suppress carcinogenic potential in cellular and animal studies.
So DSG3, or desmoglein 3, is a member of the desmoglein and cadherin cell adhesion family. And typically where you would see this gene in action is if you had a blister, it's involved in the healing process and helping your skin go from a cut or a boil to back to normal.
DSG3 can also bind to calcium and help form junctions or connections between cells, and is involved in attaching to intermediate filaments that monitor cell-to-cell adhesion. DSG3 may have many different roles in cell adhesion, which is why it is interesting to focus on in cancer cells.
One of the papers we read about for this gene is how it's overexpressed in head and neck cancers, which is why it could be a potential target for preventing metastasis or spreading of cancer.
The paper concluded that DSG3 was overexpressed in around 61% of the patients, and inhibiting it significantly decreased cell growth.
After researching DSG3 through various papers, we investigated it further through the bioinformatic databases KM plotter and CBio portal. We were able to set different parameters and select different samples of breast cancer patients to see if the high expressions of DSG3 affected patient survival.
Based on the plot we created using CBio portal, there's a statistically significant difference between patient survival in patients with high versus low expression of DSG3. Bioinformatics databases are useful to look at before spending time and resources on a lab experiment to see if the hypothesis is worth investigating.
As we conclude this final episode of our video series, We want to say thank you to everyone who has helped us throughout the year on this project, as well as everyone who's watching our videos. We couldn't have done it without your support and help.
First, we want to give a special thanks to Miss Doss for directing the Fellows Program and keeping us on track throughout the year. Second, we want to thank our wonderful lab mentors, Megan and Julia, for teaching us everything about lab techniques and taking the time to answer our questions about anything. We seriously would not have been able to do this without their expertise and guidance. Next, we want to thank our spectacular mentors, Dr. Stewart and Agina Martin, for providing us with this amazing opportunity and providing us with a lab space and mentors to learn from. Finally, thank you so much to the Eni family for this opportunity to pursue this year-long project.
So, thank you to everyone who has tuned in. We've learned so much throughout this project, and we're so incredibly grateful for this experience.